Hello everyone, welcome to day 11 of genetics. Today we are going to discuss the chromosomal disorders. Before going into the chromosomal disorders, you should know what do you mean by karyotyping. Karyotyping, it is a basic tool to study almost all the cytogenetic abnormalities. Okay, it is a basic tool to study almost all the cytogenetic abnormalities. So before that, in which phase of the cell cycle we are going to study karyotyping? The answer is metaphase, because in metaphase all the cells will be arranged in the equator. Am I right? Yes. And there will be formation of what? Mitotic spindle. In order to prevent the cell from progressing to the other phases of cell cycle, that is, we have from metaphase to anaphase, we have to use some colchicine. What is colchicine? Colchicine is nothing else but a mitotic spindle inhibitors, so that what will happen in these cells will remain in the metaphase and we can understand the morphology of these chromosomes very well. So the basic structure of the chromosome. I hope the chromosome has a centromere and it has what we call it as 4 chromatids. Okay, and one we call it as the short arm of the chromosome and another is what we call it as the long arm of the chromosome. Short arm is called as the P arm and the long arm is called as the Q arm. P stands for petite. Okay, yes. And remember, we have various staining methods. Okay, we have various staining methods. Out of that, the 2 important stains, just you have to know the name of these staining methods, because that is the one which will be asked in the MCQ. One is what we call it as G banding. G banding stands for Giemsa banding. Okay, G stands for Giemsa. And another is what we call it as Q banding. Q stands for quinacrine. Okay, Q stands for quinacrine. Okay, that is very, very important. Okay, these are the 2 important points we have to remember. Okay, see, this is the Giemsa banding of a karyotype. See, we have an arm we call it as the P or Q. Then we have a region, we have a band, and finally we have a subband. For example, I am giving you an example. Okay, that is XP21.2. Okay, we can read like that. So X stands for the chromosome, X chromosome. That is the P stands for short arm of the X chromosome. 2 stands for region. Then band is 1, and finally subband is 2. You should know how to interpret a chromosome, because and there is no need that you should remember in depth about that. Is only in some cases. For example, in case of cystic fibrosis, in that is CFTR, only in some cases you have to remember this the band and subband regions. In other cases, there is no need that you have to memorize the whole stuff, but you should know what does each letter represent in a karyotype. Then we have to see about what are the structural abnormalities of the chromosomes. Okay, we have what we call it as euploidy. Euploidy is nothing else but what? Any exact multiple of the haploid number of chromosome, we call it as euploidy. For example, 23. For example, if it is not an exact multiple of 23, we call it as aneuploidy. And remember, there are 2 important reasons for aneuploidy that they may ask in MCQ. One is called as non-disjunction of chromosome and the other one is what we call it as anaphase lag. So remember, aneuploidy, the 2 important reasons are meiotic non-disjunction and the next one is called as anaphase lag. Just to remember the key points. There is no need that you should know in depth about the mechanisms and all. Okay, yes. So whenever there is this mitotic errors, okay, what will happen? It will give rise to different chromosomes in the same individual, and that is what we call it as mosaicism. What do you mean by mosaic? Mosaic pattern. Mosaic pattern. That means you have seen in olden houses and all, you will have some special tile which will be having what we say mosaic pattern. Am I right? Mosaic pattern. So what is this mosaic pattern? In the same tile, that means in the same individual, what there will be different chromosomes. So there will be different chromosomal abnormalities, and at the same time, what will happen? This will rise to different phenotypical features. So this is what we call as mosaicism. Okay, yes. So remember, mosaicism can be what occurs as a result of any problem in the fertilized ovum or in the somatic cells. Before the chromosome fuse itself, they can have problem, or after they give rise to zygote also they can have problems. Okay, fine, yes. So what are such mosaicism? That is what we are going to discuss. Turner syndrome. Then we will have what? Klinefelter syndrome. Okay, all those yes, that is what we call it as mosaicism. Okay, all those we will be dealing in detail. Okay, so what do you mean by mosaicism? Means they are not normal. Just remember this very basic point. That means the karyotype is not normal. For example, in Turner syndrome, we have what is the karyotype? 45XO. In Klinefelter syndrome, it is 47XXY. Am I right? Yes. So these are some of the important abnormalities. Okay, yes. Okay, once that refer, let us know some of the important terminologies. Okay, what do you mean by that means some of the commonly encountered terminologies that we came in genetics. Okay, number 1 is called deletion. So in the name itself, we can indicate deletion means what? Deletion means what? Yes, a part of the chromosome, am I right, is undergoing breaking and that is undergoing deleted. So the best important example you have to remember in deletion is Cry-du-Chat syndrome. Cry-du-Chat syndrome, in which there will be a short arm of chromosome number 5 will undergo deletion. Okay, that means what do you mean by cry do chat? That means the baby will be having cat-like cry. That is cry do chat syndrome. This is the only one example you have to remember as far as deletion is concerned. Then what do you mean by ring chromosome? See, they will ask the straight away MCQ questions. Okay, in your prof exams you will be getting. That means when for example, this is a chromosome. Okay, this is a chromosome, and when the break occurs at the ends of the chromosomes, and I will show the picture also. 1 second. What is isochromosomes? What is ring chromosome? See, the break occurs at the ends, and finally they will fuse in the form of a ring, fuse to form a ring. This is called as a ring chromosome. Okay, yes. Then what do you mean by inversion? Inversion means what? Inversion may be either a paracentric inversion or a pericentric. Pericentric means what we call it as involving the centromere, and paracentric inversion means without any centromere inversion. Okay, I will show the example for that also. See, this is when it occurs on the sides of the centromere, we call it as paracentric, and when it occurs including the centromere, we call it as the pericentric. Okay, yes, all these are types of chromosomal rearrangements. What do you mean by isochromosomes? Isochromosomes. Okay, yes. See, what do you mean by iso? Iso means what? Same. That means because of some problems, what happened? One arm of the chromosome will be lost. Okay, for example, this is the chromosome. It is having what? 2 short arms and 2 long arms. Okay, suppose what will happen? One arm will be lost. Okay, this arm will be lost. Imagine that this arm will be lost. So what will happen? The other end will be undergoing duplication. So finally the chromosome will be having either 2 short arms, okay, or the chromosome will be having 2 long arms. Okay, this is called as iso. Iso means same. So one arm is lost, the remaining arm will undergo duplication. So finally the chromosome will be consisting either 2 short arms or either 2 long arms. Okay, yes. And remember a very, very important question in isochromosome, that is the genetic information which present in both arms are morphologically identical, whereas that is not the case with other arrangements, that is in ring chromosome or inversion, that is whether it is paracentric or pericentric, or in case of translocation there is alterations of the genetic information. That means the genetic information is not identical as that of the parent chromosome, whereas in case of isochromosome the genetic information is identical. This is the very, very important point. I have marked it out here as very, very important. Okay, yes. So how will you identify it is an isochromosome? In that you will be having the letter I, small i. Small I in this indicates that it is isochromosomes. Fine. Then we have translocation, which is of 2 types. One is called as balanced translocation, and one is called as Robertsonian translocation. Okay. Okay. So what do you mean by balanced translocation? What do you mean by balanced translocation? Let us see. In case of balanced translocations, what will happen? There will be single break in each of the 2 chromosomes with the exchange of materials. Whereas in case of Robertsonian translocation, what will happen? It is Robertsonian translocation, that we use that terminology especially for acrocentric chromosomes. Okay, acrocentric chromosomes. So in case of normal, or what we call it as balanced translocation, see, as you can see here, as you can see in this picture, there will be a single break. Whereas in case of Robertsonian translocation, see, first of all, remember the Robertsonian translocation is mainly, or it takes Robertsonian translocation occurs only in case of acrocentric chromosomes. In acrocentric chromosomes only the balanced translocation takes place, whereas Robertsonian translocation takes place. That is not the case with the balanced or reciprocal translocation. Fine. Yes. So into acrocentric chromosomes, we will have this Robertsonian translocation, and here the break occurs close to the centromeres of each chromosome, whereas in case of balanced translocation it occurs at the ends almost. Okay, yes. So finally what will happen? This will leads to a very large chromosome and an extremely very small chromosome. See here, you can appreciate in balanced. Why it is called balanced? The final chromosome forms are almost same in size. Fine. Yes. Whereas in case of Robertson translocation, one is extremely long and another one is extremely small. Okay, yes, and this extremely small chromosome may be lost and it won't have any function at all. Okay, yes. And the best example, of the Robertsonian translocation is in Down syndrome. Once we comes in Down syndrome, we will say in Down syndrome, don't just remember that it is just trisomy of the 21st chromosome. We have lot of abnormalities. It may be due to meiotic disjunction. Okay, and we have the role of Robertsonian translocation there. All those they are there. Okay, we will come to that. Fine. Okay, then coming into the Down syndrome, which is nothing else but called as trisomy 21. So I'm just going to say only the key points. Okay, rest all clinical features, everything is your part. You have to just by heart it. Just I'm going to say the key factors that you have to mention when they ask Down syndrome as a short note question. So first of all, remember it is the most common of the chromosomal disorder, and it is a major cause of mental retardation. Okay, it is a major cause of mental retardation. So the most common hereditary or inherited cause of mental retardation question? The answer is Down syndrome. What is the second causes? Fragile X syndrome, that we will come later. Okay, and remember the maternal age has a strong influence on the incidence of trisomy 21. That is the very first important. Okay, yes. So the meiotic non-disjunction of chromosome number 21, which occurs in the ovum. And another one important thing, there is a Robertsonian translocation involving what are the two chromosomes? Yes, what are the two chromosomes? Very, very important. There is Robertsonian translocation which occurs between chromosome number 21 and chromosome number 21 and chromosome number either 22 or 14. Okay, because translocation can occur only between two chromosomes. Am I right? Yes. So just remember that one point. And another one important point is that this Down syndrome 1% have what? Mosaics. That means a mixture of either 46 or 47 chromosomes. Okay, yes. So these all points you have to remember. So what are the genetic abnormality? It is not just simply the trisomy of 21. Okay, number one, maternal age has an influence. Number two, Robertson translocation between chromosome number 21 and chromosome number 22 or 14. Number three is mosaics, that is with either 46 or 47 chromosomes. And then we will come to the clinical features. See, these are the clinical features of Down syndrome. You will have mental retardation. All this you have to by heart. Epicanthic folds, simian crease, mental retardation, congenital heart defects, intestinal stenosis, umbilical hernia, predisposition to leukemia and hypotonia. Okay, and what is the most common type of leukemia we are getting is ALL. Okay, it is ALL is the most common type of leukemia in Down syndrome. What is the most common type of AML in Down syndrome? Answer is AML M7. Okay, that you will learn in hematology. Okay, what is AML M7? That is megakaryocytic leukemia, acute megakaryocytic leukemia. But what is the most common? Most common is still ALL. No controversies. Fine. Then hypotonia, gap between the first and second to all those features you can see here. See, you can see trisomy, translocation, and finally mosaicism between 46 or 47 chromosomes. Okay, I hope things are very clear for you. Yes, then now let us go into some other important points. See, most commonly, what are the congenital cardiac defects? You will have ostium primum defects, atrial septal defect, VST, okay, atrioventricular valve malformations. Then remember, they have either 10 to 20-fold increased risk of developing acute leukemia. Out of that, acute lymphoblastic leukemia is most common, and among the acute myeloid leukemia, acute megakaryocytic leukemia or AML M7 is more common. Then of course they will have what autoimmune disorders, okay, predisposed to a lot of autoimmune disorders also. Okay, then once that is done, and now let us see the various other trisomies. That is Edwards syndrome, and which is called as trisomy 18, and Patau syndrome, which is trisomy 13. Okay, Edwards syndrome and trisomy 13 and trisomy 18. Okay, now let us see about first Edwards syndrome, then we will come to Patau syndrome. Okay, this is Edward. Okay, this is Edward, in which there will be a prominent occiput, micrognathia, low set ears, overlapping fingers, rocker bottom foot. Remember, rocker bottom foot is present in both Edward and Patau syndrome. Please don't differenchiate both of the syndromes with these features. Then you have to just remember this micrognathia. Okay, yes, micrognathia, prominent occiput. Whereas microcephaly, see, microcephaly is a feature of what we call as Patau syndrome. Polydactyly, umbilical hernia, see, cleft lip, cleft palate. Okay, that is very important. Cleft lip, cleft palate is present in Patau syndrome, whereas it is absent in case of Edwards syndrome. Okay, yes, just go through these features. Then we have another important thing, 22q11 deletion syndrome. That means deletion. Okay, deletion I have told you, one example, what is that? Karyotyping syndrome. Similarly, on the long arm of the chromosome 22, there will be a deletion of the band q11.2. So that is why, which causes what we call as DiGeorge syndrome. What is it called as? DiGeorge syndrome. I will tell you a mnemonic to remember the features of DiGeorge syndrome. CATCH. Okay, CATCH. What do you mean by C? C stands for cleft lip and cleft palate. Cleft lip and cleft palate. Okay, then ATCH. Okay, then what to say? Okay, yes. Then T stands for thymic aplasia. Thymic aplasia. Then C stands for congenital heart defects. Congenital heart defects. And H stands for hypocalcemia. Hypocalcemia. And one more thing, that is A stands for abnormal phases. Abnormal phases. So this is CATCH. You can remember this by mnemonic CATCH. Okay, yes. So whenever they give a clinical history, or you can just remember the clinical features of this, remember hypocalcemia. Why this hypocalcemia? Because there is a defect in the formation of the third and fourth pharyngeal pouches. So as a result, the parathyroid gland and the thymus won't be produced, but there will be normal thyroid. Thyroid will be there, but the parathyroid and thymus glands won't be produced. Okay, yes. So there will be what? T cell immunodeficiency. To T cell immunodeficiency, that is very, very important. The B cells is normal. The cell mediated immunity is defective. Okay, the cell mediated immunity is defective. That is the first important point you have to remember. So on histopathology, on HPE, histopathological examination, what you can see decreased size of paracortical areas. This is a very commonly asked question. Decreased size of paracortical areas of lymph node. But if you know the basics, you can very answer this type of question. Why? Because in the paracortex of the lymph node only we have T cells. Since in DJO syndrome the T cells of the cell mediated immunity is defective, there will be decreased size of paracortical areas. Okay, I hope this is the histopathological examination finding. Okay, yes. Okay, that's about that. Okay. Then once it is done, DJO syndrome, and remember DJO syndrome, DJO syndrome. There is one important gene which comes in the pathogenesis of DJO syndrome. That is what we call it as TBX1. Okay, TBX1 gene, and another one is called as PAX9. Okay, TBX1, it is a transcription factor. TBX1 is a transcription factor, and the gene involved is PAX9. TBX1 is a transcription factor, and the gene involved is PAX9. Okay, yes. So because these genes are mainly involved in the development of the palate, parathyroid, and thymus, it has been once asked in a Central Institute exam. Fine. Next is another one important thing. What do you mean by Lyon's hypothesis? Just simply I am telling Lyon's hypothesis, if the two X chromosomes are there, one X chromosome will undergo inactivation, and the another one X chromosome only will remain active. So the inactive X chromosome, now that is what we call as the bar body. So how will you calculate the number of bar body? Number of bar body is equal to the number of X chromosome minus 1. Simple. So, and remember the molecular basis of this X chromosome inactivation includes a long non-coding RNA. Already I have mentioned it several times in the first chapter, LNCR, long non-coding RNA. What is the name of this long non-coding RNA? The name of the long non-coding RNA is called as the cyst gene. Okay, what is the name of this gene? Called as cyst gene. Okay, this is a long non-coding RNA. Fine, yes. Then we have something what we call as the Klinefelters. Okay, yes. So you know the SRY gene. The SRY gene is nothing else but the sex determining gene which is located on the short arm of the Y chromosome. Okay, this is responsible to it is this chromosome, or it is this gene which decides whether the baby should be born male or female. Suppose if this SRY gene is absent, the baby will become female, and if it is present, the baby will become male. Okay, just remember this point. Then Klinefelter syndrome, as well as Klinefelter syndrome is important. Okay, Klinefelter syndrome and Turner syndrome, right? Yes. In Klinefelter syndrome, you know, the IQ will be lower, okay, and sometimes even mental retardation may be present. Then you just go through the other features like what atrophic testis. You know what is the chromosomal abnormality in case of Klinefelter syndrome? It is 47XXY. Okay, already you have learned Klinefelter syndrome and all in your lower classes. Okay, yes, just go through this important. And what is the histopathological examination finding? Okay, histopathological findings in Klinefelter syndrome, that is the seminiferous tubules will be atrophied. It will be replaced by pink hyaline collagenous ghost. Okay, yes, and there will be atrophic tubules. Then they have increased risk of breast cancer, extragonadal germ cell tumors, autoimmune diseases like systemic lupus erythematosus. 47XXY is the karyotype. Fine. Okay. See, this is Turner's and Klinefelters. Okay, next we will go for Turner's. So as far as Turner's is concerned, just remember what it is, hypogonadism. Okay, hypogonadism in phenotypic females. Okay, it is a partial monosomy, 45XO, partial monosomy of the X chromosomes. Then the common feature is what? Partial monosomy. So the main structural abnormality is partial monosomy of the X chromosome. Then another important, just I am going to highlight only this, and remember this, the patients are at higher risk of development of gonadal tumor or gonadal blastoma. Then they have some important characteristic features that you may miss. That is, number one is cystic hygroma. Okay, then webbing of the neck. Okay, short stature. Okay, short stature, and the most important cardiovascular anomaly is bicuspid aortic valve and coarctation of aorta. Bicuspid aortic valve and coarctation of aorta. Okay, yes, little pubic hair. Okay, that is failure to develop. That means secondary sexual characteristics will be absent, and the most important cause of primary anuria is actually the Turner syndrome. Okay, yes. and they will have streak gonads or the streak ovaries. But remember, the streak ovaries have, unlike the testis, okay, unlike the testis which have increased risk of gonadoblastoma, the streak ovaries do not have risk of gonadoblastoma. Okay, fine. And the most important gene which is mutated here is Shox gene, which is the short stature homeobox gene. Because of this gene mutation only, the females are short stature. Then another important is cystic hygroma. Okay, see, all features are here: short stature, coarctation of aorta, low posterior headline and webbing of the neck, broad chest and widely spaced nipple, pigmented navy, streak ovaries, infertility, amenorrhea, and peripheral lymphedema at birth. Okay, yes. Okay. Once this is done, next we will go for what we call it as tri-nucleated repeat mutation. Out of that, we have, remember, as far as tri-nucleated repeat mutation, that means a codon, a particular codon is undergoing repeated many times. Okay, that is what we call tri-nucleated repeat mutations. We have some diseases which affects the non-coding regions and some which affects the coding region. Just remember the diseases which affects the non-coding region. Rest all you can write, they are coding regions. Fragile X syndrome, Friedreich's ataxia, and myotonic dystrophy. So these are the three conditions which affects the non-coding regions. Fragile X syndrome, we have CGG. Okay, ataxia means you have 2A, so it is GAA. And finally myotonic dystrophy is CTG. So these are the repeats which are undergoing, or these are the codons which are undergoing many times repetition. So that is responsible for the clinical manifestations of this condition. We have Fragile X syndrome, Friedreich's ataxia, and myotonic dystrophy. So what is the gene located? Fmr1, then FXN, and DMPK. Okay, yes, that is myotonic dystrophy protein kinase, then frataxin in case of Friedreich's ataxia, and FMR, that is familial mental retardation 1 protein in case of Fragile X syndrome. Then spinobulbar muscular atrophy, that is Kennedy's disease, Huntington's disease, spinocerebellar ataxia, all type 1 to 7, all those affects the coding regions. And what is the mutation in all, what is the repeat in all of this condition? That is CAG. Out of that, spinobulbar muscular atrophy or Kennedy's disease, okay, this involves the X chromosome. It involves X chromosome, okay, and the receptor or the gene which is undergoing mutation is androgen receptor. There is no need that you have to sit and memorize the rest of the genes and chromosome numbers of all other conditions. Fine, just remember these things. Okay, and so Fragile X syndrome, so you can see here, no? Okay, yes. See, Fragile X syndrome is there, as well as we have Fragile X tremor ataxia is there. So what is actually the difference between Fragile X syndrome and Fragile X ataxia? In Fragile X syndrome, there is a loss of function mutation, whereas in case of Fragile X tremor ataxia, there will be a gain of function mutation. So here there is a loss, there will be problem. There, because of the gain, there will be accumulation of toxic mRNA that is responsible for the ataxia. Fine, yes. So the morphological hallmark of all these is what? There will be some abnormal proteins or the mutant protein. This will undergo what? Accumulation in the form of large intranuclear inclusion bodies. Fine, yes. Okay, once that is done, let us go for Fragile X syndrome. Fragile X syndrome, I told you, the Down syndrome is the most common genetic cause or the hereditary cause of mental retardation. After Down syndrome, it is the Fragile X syndrome, it is the most important cause. It is caused by a tri-nucleated mutation in the Fmr1 gene, that is familial mental retardation 1 gene. Okay. So why it is called as fragile? Why? Because the cytogenetic alteration. So when they stain, I told you, we use Jimsa or Quinacre staining. In staining, they found that the long arm of the X chromosome, there was a constriction. So it appears as if the chromosome was broken, and hence it was called as the fragile site. Okay, and remember, they are mentally retarded. They have pure IQ, that is 20 to 60. Okay, and what are the classical features of Fragile X syndrome? Long face, large mandible, large testis, large ears. Okay, yes, all these are the features of Fragile X syndrome. Okay, yes, and the most distinctive feature of Fragile X syndrome, is macroorchidism. Okay, the most distinctive feature of Fragile X syndrome is what we call as macroorchidism or large testis. Okay, and fine. So next is, what is the gene involved? It is the Fmr1 gene. I told you what is the repeat. It is CGG. And remember, what do you mean by pre-mutations and what do you mean by amplification? So these are some of the important things. And yes, so let us see. So I told you, these repeats. Suppose if these repeats are between 55 to 200 repeats are present, we call them as pre-mutation. And 200 to 4000 repeats, we call them as full mutations. Fine? Yes. Okay. Yes. And okay, fine. Yes, what do you mean by anticipation? The phenomenon of anticipation is a characteristic. Anticipation can be asked as a what MCQ question. Okay, anticipation is one. What do you mean by anticipation? That means there will be exaggeration or there will be worsening of the clinical manifestations in subsequent generation. That means as the generation progress. For example, this is a father and a mother. This is a father and a mother. They have one children. This children is going to have another two children. Okay, and this boy is going to have another. So this child will be the worst affected. Why? Because of anticipation phenomenon. There will be amplification of the mutant genomes in successive generation. This phenomenon is called as anticipation. So there will be worsening of the clinical conditions as the generation progress. Okay, this is what we call as anticipation phenomenon. Okay, yes, that means the great grandsons will be the most which is going to be affected in this condition. Okay, yes. Okay, then once this is done, just remember a very important point. Just remember that 55 to 200 and 2000 to 4000 repeats. Okay, once this is done, let us go for the another condition that is called as I told you what is FMRP. What is FMRP? It is familial mental retardation 1 gene. And what is FRMP? FRMP is nothing else but it's a translation regulator. Okay, FRMP is nothing else but a translation regulator. Just remember that. There is no need that you have to remember the rest of the things. Okay, what is Fragile X tremor or ataxia? I told you that here the mutation is also in Fmr1 gene, but how it differs from others? The Fragile X syndrome, it is a gain of function mutation. Okay, it's a gain of function mutation. Okay, what is the same? The repeats are again same. That is nothing else but CGG repeats. Okay, it is CGG repeats. Okay. Next, mitochondrial disease. Out of that, the most important point you have to remember in case of mitochondrial conditions is Leber's hereditary optic neuropathy. We have lot of disorders which comes under the mitochondrial inheritance. That is what MELAS. We have Kane-Sarri syndrome. Lot of Pearson syndrome. We have lot of disorders which comes under mitochondrial inheritance. Just remember, maternal DNA or maternal inheritance is a unique feature of mitochondrial disorders. Okay, only the female can transmit the disease. Okay, yes. Okay. Leber's hereditary optic neuropathy. See, it is nothing else but a neurodegenerative disorder. Okay, there will be progressive bilateral loss of central vision. Finally, blindness will be there. Even cardiac conduction defects and minor neurological abnormalities will be also present. Okay, then coming into what do you mean by heteroplasmy? That means in mitochondrial disorders, the individuals will have both the wild as well as the mutant type of DNA. Since the two different types of mitochondrial DNA is present, that condition is what we call it as heteroplasmy. Okay, yes. And remember, for the disease to undergo manifestation, only a minimum number of this mutant mitochondrial DNA must be present. So that is what we call as a threshold effect. Threshold means a minimum amount, right? Yes. So the minimum number of mitochondrial DNA is required for clinical manifestation. What do you mean by genomic imprinting? Imprinting means inactivation of alleles. So either the maternal allele or the paternal allele will be inactivated, and another that is called as maternal or paternal silencing, transcriptional silencing. Maternal imprinting, for example, I am telling it is maternal imprinting, or I am saying paternal imprinting. Maternal imprinting means transcriptional silencing of the maternal allele. Okay, paternal imprinting means the paternal allele will be silenced or inactivated. Fine, yes. So we have two conditions which comes under this. One is called Prader-Willi syndrome, and another one is called as Angelman syndrome. First, I am telling Prader-Willi syndrome. So Prader-Willi syndrome. Okay, Prader-Willi syndrome. Okay, yeah. So in Prader-Willi syndrome, what are the characteristic features? Mental retardation, short stature, hypotonia, profound hyperphagia, and hypogonadism. Whereas in Angelman syndrome, there will be ataxic gait, seizures, and inappropriate laughter, or they are also called as happy puppets. Let us see what is the molecular problem in case of this. Okay, yes. So yes, P for P, just remember like that means in paternal means what? Prader-Willi syndrome. So in Prader-Willi syndrome, okay, there will be what? Inactivation of what? The paternal chromosome. So in Prader-Willi syndrome, there will be paternal imprinting. I can also say, so just remember this point. In Prader-Willi syndrome, the paternal allele is silenced. So the maternal allele will undergo duplication, or it will undergo amplification. That is what we call it as paternal silencing with the maternal disomy is Prader-Willi syndrome. Similarly, in maternal, okay, that is in angels, okay, remember like mothers are angels. So in Angelman syndrome, there will be maternal silencing and paternal disomy. Okay, this is a very important thing, just simple only. Okay, yes. So the Prader-Willi syndrome, they will have what? They will have two maternal copies of what is the chromosome which is involved here? Chromosome number 50. Similarly, Angelman syndrome have what? Two chromosomes. Okay, that is disomy of the paternal chromosome. Okay, so this is the most important thing. And one important gene, okay, which is expressed here is nothing else but the UBE3 gene. Okay, what is the name of the gene called? UBE3A gene. Just remember the gene. Just remember what is the problem and what are the clinical features. There is no need that. Okay, yes. Suppose if you want to remember a little bit more specific, it is 15q12. It is 15q12. Okay, just to remember this point. Okay, yes. Okay. Okay, so what is that? UBE3A. Okay, these are the important points you have to remember there. Once this is done, let us go for what do you mean by gonadal mosaicism. See, in gonadal mosaicism, what is actually happening? Gonadal mosaicism means, in some conditions, like predominantly osteogenesis imperfecta, the parents will be phenotypically normal, but the same parents have affected child. So what is the mechanism behind that? That means the mosaicism results from a mutation that occurs post-zygotically. And if this mutation affects only cells destined to form the gonads, the gametes will carry the mutation, but the somatic cells of the individual will be completely normal. So just remember, children. So the parents are normal, but the child is affected. How is it possible? It is the sex chromosome, am I right? Which takes part in the fertilization and all those stuffs. So when there is any problem, and remember these changes will take place post-zygotically, after the zygote is formed. So suppose if this mutation is going to affect this sex chromosome cells, only the problem will be there. That means these gametes will carry the mutation, and as a result they will causes the child to be affected. That is the reason for the child to be affected, or for an affected child. And so at the same time, when this problem is carried by some somatic cells, there won't be any problem. So what do you mean by gonadal mosaicism? Phenotypically normal parents have affected child. What is the mechanism or why it is happening? Because the genes will be carried out by gametes. Then the child will be affected. Suppose if the genes are carried by the somatic cells, no problem. The child will be normal. So what is the characteristic disease? A single disease you have to remember here, that is osteogenesis imperfecta. Okay, just remember, gonadal mosaicism may be asked in an MCQ question. Okay, sometimes, very rarely it may be asked as a short note question. So just remember, Prader-Willi syndrome, mental retardation, short stature, hypotonia, hyperphagia, small hands and feet, hypogonadism. Angelman syndrome, we have what we call as mental retardation, ataxia, inappropriate laughter. That is why they are called as happy puppets. Okay, that's why they are called as happy puppets. Okay, I hope you are clear with all these points. Yes, good. Okay, so with this, we have finished this. Then we have one more session which is pending in case of genetics. That is nothing else but what we call as the molecular diagnostics parts of genetics that we will cover in another session. Hope you have enjoyed this session. Thank you.